antibody-based rapid detection of carbapenem-resistant accelerated vector dominant, advancing global health and antimicrobial cures. So, as ma'am has already explained in her lecture about the antimicrobial resistance, so I will not be going to the brief of the antimicrobial resistance. So, the need of the R, as we are targeting the development of protein-based reagents for the detection of carbapenem-resistant bacteria. So, carbapenem-resistant pathogen Acinetobacter baumannii has been listed as critical priority pathogen 1 by WHO in 2024. So, what are carbapenems? Carbapenems are third generation antimicrobial agents which belongs to beta-lactam antibiotic class. They work by inhibiting the synthesis of bacterial cell wall. So there are multiple mechanisms due to which the carbapenem attain the resistance against the bacteria attains the resistance against the antibiotic. So we are focusing on the expression of carbapenemases. These are the enzymes which degrade the antibiotic. So the need of the R is the development of protein-based reagents for detection of carbapenem-resistant bacteria. So the studies suggest that carbapenem resistance in Acinetobacter baumannii, it is mediated by carbapenemases. So most of the currently available methods, these are expensive, laborious, and time consuming. So our aim is to develop antibody-based immunoreagents for the detection of carbapenem resistance in Acinetobacter baumannii. So we have chosen a major carbapenemase as a target for development of antibody-based immunoreagents, and it would be useful for the timely treatment of infected patients, and it will improve the clinical outcome that will allow the controlled use of carbapenem drugs and effective management of infection spread. So coming up to the methodology, so our first target was to clone the key carbapenemase, express it and purification, then followed by immunization of the antigen, the carbapenemase, into the mice. Then after proceeding it for the complementary DNA, then the amplification of variable light chain and heavy chain gene. So we are using fast display technology for the development of antibodies. So after cloning, after the amplification of variable light and variable heavy chain region of the antibody, we assembled it as STFB by linking it to a linker. Then after cloning it to a fast display based vector, we found the combinatorial STFB library. Then for the selection of specific clones against the antigen, we performed the 2 rounds of biopanning in which the unspecific ones are washed out and the specific ones are collected separately. Then we got the healthy STFB library, and there are next steps were the conversion of it to the antigen, it to the desired format, which may be STFB or STFB format. Then we tested with the transformation to a assay kind of set or collection. So our first step was the purification of the key carbohydrates which we have targeted. So we cloned it in Cat28 vector and then we transform it to synthesized cells. Then there is an NPS analysis for the different stages of purification of the carbapenemase which we have targeted. Then we purified it using nickel affinity chromatography. Then we treated the purified protein with the H6 series protein to remove the N-terminal effect. So we use this series type for the purification purpose. So for the removal of it, we treated it with H6 series protein. So our next step was the characterization of purified protein with the key carbohydrates. So to check that whether the histidine side and whether the histidine side has been removed or not, so we performed an indirect ELISA and Doppler method in which we coated the carbohydrates before the removal of the N-terminal effect and after the removal of the N-terminal effect, and in both the results, the purified carbohydrates protein did not show reactivity against the anti-antibody ELISA as well as in Doppler assay, which states that the histidine side has been removed. So our next step was the mice immunization and the development of antigen antibody. So we immunized with last 2 mice, and then after certain boosters of the antigen which we injected into the mice. So after these 2 boosters, we took the serum culture of the mice to check whether the mice are showing reactivity against the antibody or not. So we performed an indirect ELISA. We coated the serum on the ELISA plate, and then we coated the key carbopenemase on the ELISA plate, and we added the serum which we got from the mice, and then we detected it with secondary antibody, and in results 4 to 5 mice were showing good immune response against our purified recombinant carbapenemase protein. So our next step was development of fast display SCFP fast display library again carbapenemase protein. So we amplified the variable light chain and variable heavy chain region which are responsible for the binding of the antibody to the antigen, and then we assembled it as a assembled it as SCFP single chain fragment variable. Then we cloned it into our fast display based vector, and then we formed our library of 10 to the power 6 transformants. Then for the selection of specific clones, we performed 2 rounds of biopanning, and then after selecting 6 clones and confirming it with the help of sequencing, we select 6 clones and we performed indirect ELISA for selection of specific antibodies and their screening. So we coated our carbapenemase and E. coli lysate, which was the host for the production of the carbapenemase. So all the 6 clones, they were showing reactivity against only our carbapenemase protein, not with the E. coli lysate, which was the non-specific study. Then we analyzed the selected antibodies in the soluble form to check whether their activity is still intact or not. So we performed a fast ELISA in which we coated the key carbapenemase on the plate, and then we cloned the SCFPs into a biotin-related vector, biotin acceptor peptide vector. And then we detected this streptavidin HRP, and in the result, the results were same. All the 6 clones were, their activity was still intact and they were specific against the target carbapenemase. Then our next step was to check the SCFP that the minimum detection limit of the bacterial protein in a sample. So we performed our dot plot for this test to check the minimum detection limit. So we coated different we added different drops of the SCFPs, different drops of the carbapenemase on the strip, and then we checked nitrocellulose membrane were used for the dot plot assay, and in our result we got that our developed reagents were able to detect 6.25 nanogram per ml of the bacterial protein in a sample. And right now we are in a process of developing a lateral flow assay kind of test for rapid detection of antibiotic resistant bacteria. Coming up to the conclusion, so our research employs the fast display technology for develop monoclonal antibodies using major carbapenemase as a primary target, which is a key factor for the resistance in the accelerator vector bomb-like. So these antibodies will be characterized further, and selected binders will be used for developing point-of-care assays for profiling antibiotic resistance in the drug-resistant accelerator vector bomb-like. So coming up to the acknowledgement, first of all, I want to acknowledge my supervisor, Dr. Vaishali Verma, for her invaluable guidance and support, Bennett University for the